This lecture really consists of genetic material, really talking about DNA. Here, RNA will definitely come into play as we talk about the central dogma. So first I'm going to really talk about what DNA is, going back to that, a little bit of history. Watson, Crick, and Franklin come into play here, and I will end the lecture on the process replicating DNA and what enzymes are involved in that beautiful, very elegant, very detailed process that our genetic material replicates quite efficiently, and it does this pretty quickly and consistently. So we'll talk about that. Our next lecture then goes on and continues the central dogma from genetic material, from nucleic acid to protein. So really what DNA is doing, and it holds genes, these genes encode for a protein. They give that blueprint for that end product that's going to contribute to structure and function of an organism, talking about protein. So DNA, you've heard me talk about most of these terms that I'm going to throw out. Really, the chromosome is what we're getting towards. And I really want to teach you exactly what a chromosome is. So there are different ways that we can picture DNA. Here I see the DNA double helix. This is what you guys have learned. There's Watson and Crick's beautiful model, DNA double helix. That's very much how DNA is. But DNA is quite long. It's millions of nucleotides long. And so what ends up happening is that DNA kind of gets packed up and bunched up and very, very dense, really, really coiled together in order to make a chromosome, which is what you guys saw in your onion cells during the mitosis lab. Very, very densely packed DNA. So let's go through this together. The DNA in a chromosome is packed into a very elaborate multi-level system. This is different coiling that occurs. You guys are a little bit young, but perhaps you have seen or heard maybe of telephones having cords back in the day, and that cord had a little coil, maybe like a little pig's tail as well. That's a coiling effect that's going on, a little wound effect. That's very much how DNA is packed. And then DNA also folds on itself as well. So as we see this folding occurring, we actually see a new player coming into DNA called histones, these little purple circles right here. Histones are proteins. So they're not nucleic acid, more so than they are proteins, very much a part of DNA. They help package the DNA in eukaryotes. I can kind of see here multiple histones kind of coming together so that the DNA is able to wrap around those proteins. And it really helps with this coiling effect that's going to pack the DNA even further. We refer to these histones and the DNA that wraps around them as nucleosomes. Here we can see one nucleosome, multiple histones and DNA coiling around it. That's called a nucleosome. I can see the nucleosomes here on this image. Kind of resembles beads on a string. So they're those tightly packed nucleosomes, those dark circles, and then we can see the DNA coiling around it and becoming that string. Looks like beads on a string as we get packed. Eventually, as we go further, we can see that those nucleosomes kind of fold in around each other. There's more coiling, coil on coil. As we are condensing the DNA, at this point, I want to throw in the term chromatin. Chromatin results when nucleosomes are packaged into a thread. So we've got proteins and DNA, and we are packing, and then coiling and looping and folding even further, and then even more to the point where we are condensing. We're tightly packing that chromatin. Remember, chromatin are nucleosomes, DNA and proteins, tightly packed to where we end up getting, oh, I don't have any text, but we end up getting a chromosome. Here's a chromosome. You guys are used to seeing that. It does look like this is a duplicated chromosome since we do have sister chromatids. Kind of have that X-like image on this chromosome. So they're sister chromatids. It's probably at the end of S phase. It can be in the G2 phase. It could be at the beginning stages of the mitotic or meiosis phase. But we have sister chromatids. There's that pinched area here that you guys have seen before. That's called the centromere. That area of the chromosome where we're going to have specific nucleic acids where the sister chromatids help attach and where the spindle fibers will attach as well. There's my chromosome. Very tightly packed. correct DNA. So if I were to have DNA like this in my cell and stretch it out into a thin string, it would be very long. In fact, so if I were to take the DNA in your cells and lay it out as a long piece of string, it would actually be about 6 feet long, give or take, roughly. That's pretty long, about 2 to 3 meters. To kind of relate this to an everyday item, that would be like the length of a dining room table. So it's just one cell, and that's how long your DNA is. So it needs to be kind of tightly packed and ordered in such a way in order to contain it. Right? So you guys have seen chromosomes. Our cells don't always have this state. The DNA is not always found in this state in cells. Chromosomes are found towards the end of interphase when DNA is getting ready to undergo cellular reproduction, so mitosis or meiosis. You saw that during mitosis for your onion cells during your lab that the DNA was quite wormy looking, very stringy looking, and quite dark compared to your interphase, your cells in the interphase. And so DNA is not always found this way.